All right, so chapter 3. Chapter 3 is going to be on bacteria and archaea. We'll go over the different structures within the cell wall. Big heavy focus on the cell wall throughout this chapter. A little bit on the cell membrane, but it'll knowing the structure of the cell wall will help us determine whether a substance is gram positive or gram negative. We'll look at some internal structures with our bacteria, and we will also look at which structures some bacteria have that others may not. They could have these extra structures which kind of give them maybe an advantage over other bacterium. And we will also look a little bit at archaea and just give kind of like a summary of why it's in its own domain and where you're most likely to find it. So as we get started with our bacteria, our prokaryote, we want to do some comparisons between prokaryotes and eukaryotes, just to kind of review from the last set of videos, the last chapter. So prokaryotes are notoriously smaller than eukaryotes, which is why we typically need a microscope to even be able to see them at all. Versus eukaryotes, we can use microscopes to see them and pull them into better focus and get a better view of them. But generally, most bacteria, most prokaryotes need to be seen with a microscope. Now, prokaryotes have peptidoglycan in their cell wall, and that's something we're going to explore more throughout this chapter, but I do want you to become familiar, with the term peptidoglycan. It's going to help it's going to help us discover the bacterium's gram status, whether it's gram positive or gram negative. And it is kind of like a defining factor for a bacteria. With the eukaryotes have a plasma membrane with that phospholipid bilayer. With prokaryotes, DNA is in that nucleoid region, which we've already explored. Eukaryotes have that nucleus where their DNA is located. Prokaryotes have ribosomes, that's about it, and they have no other or no membrane-bound organelles. Unlike eukaryotes, which have the membrane-bound organelles, which would consist of like the nucleus, the mitochondria, Golgi apparatus, endoplasmic reticulum, the list goes on, right? Prokaryotes lack those items, but they do have ribosomes, and eukaryotes have ribosomes as well. And you will see that eukaryotic ribosomes tend to be a little larger than prokaryotic ribosomes, which makes sense because eukaryotic cells tend to be larger in general than prokaryotic cells. So let's look at our different shapes of prokaryotes. This is going to be particularly important throughout the labs that we do. And it's kind of like a little bit of a different language, not too complex, just a couple of prefixes and suffixes to learn. Nothing crazy, I promise. But to start, it says up there that bacteria are pleomorphic. And you do want to know what we mean by pleomorphic. And when we say pleomorphic, it means that a single species can vary in size and shape. So you're going to see in the next couple of slides, a lot of times we'll classify bacteria as either cocci, which I'll flip to over here, cocci, meaning kind of like spherical or round in shape, right? Or bacilli, and bacilli are rod-like, kind of like the streptobacilli down here. Okay, just rod in structure. There are times when one particular bacterium could appear cocci in one setting and bacilli in another setting, right? Just depending upon its environment and maybe what the temperature is or nutritional factors that are around, maybe acidity. But bacteria can change shape depending upon where it is. Just kind of like how we tend to maybe change our looks depending upon where we are. Like I know if I'm at home, I'm going to be kind of probably in sweatpants with no makeup on. But if I'm going out to meet some friends, like in a nice restaurant, maybe I'll dress a little nicer, throw on some cover up, mascara, all that fun stuff. Okay, bacteria kind of do the same thing, but generally, you'll find that bacteria that are typically cocci, they tend to kind of remain cocci, and bacteria that are bacilli, the ones that we go to look at, they're not going to kind of change in front of you. They will also remain bacilli. But just know it's possible for them to vary in size and shape. So again, when we say rods, we mean bacilli or cylindrical or shaped like a cylinder, essentially what we have here. Couple other terms to recognize, but what I really want you to focus on is this concept of bacilli and this concept of strep, which we'll get to in a second. But they can be described as fusiform, which means that they're like a little tapered. When we say streptobacilli, we mean that it exists in chains, okay? So when you think strep, I think of steps and like kind of like walking, like kind of like in a chain-like fashion. That kind of helps me. Versus something called staph, which we'll see over here. And we kind of, I think, talked about in a previous chapter. But staph means a cluster or a bunch of grapes. And generally how a student, I'll give her credit, Brittany came up with, she likes to remember staff as like a staff meeting, like it's a cluster of people. So generally when you go to a staff meeting, it's generally, we all hate going to staff meetings, I know I do, but it's generally kind of like a group or a bunch of people when we think of staff meetings. We've also got a couple other shapes listed, and I think we'll get to Vibrio a little bit further, a couple slides down. But there's palisades, so it says cells of a chain remain like partially attached. There's Vibrio, singly occurring rods that are shaped like a comma, the structure that we have here. And then you can have these, you'll see these a lot, branching filaments where it kind of looks like trees. Generally that's strepto because they're going kind of like in chains there, but they're also forming branches as well. But your big takeaways from this slide are the concept of the pleomorphic, what it means to be bacilli, right, cylindrical, and what it means to be strep, which is a chain. Now with cocci, right, again, it means spherical, okay, and we can have some different terms to describe cocci. Cocci can be staph, right, which we said was in clusters or groups. It can be strep, again, which is in chains. It can also be diplo, right, which are pairs, right? So here's a little example of diplococci here, little pairs. They can be in a tetrad, group of 4, or they can kind of go like a little 3D and be cuboidal almost. So it says Starkina, a packet of 8 to 64 cells. And these cells tend to divide and stick together. They kind of act like a group, like a fully functional, just I guess kind of like community where they're dividing and maintaining their structure. Our third type of cell structure would be spirillum, kind of like a little spiral. You won't see too much of this in lab, but these do exist. They do look like a corkscrew. We'll come back to spirochetes with our periplasmic flagella, but it is kind of like a little coil with the periplasmic flagella that exists between the cell wall and the cell membrane. It's very interesting, actually. And it does resemble a spring. But we talked about our bacilli that can kind of form like a comma here. That's our Vibrio. And then you've got your spirillums, this like long coil, and then your spirochetes, which are a little thinner. And then we've got some comparisons here between a spirilia and a spirochete, which you are more than welcome to read through. I'm not going to go through it because it's like right there for you. Let's talk about the cell walls of bacteria, okay? It says that most prokaryotes are surrounded by a cell wall, which is super important, and the purpose of the cell wall is to protect it from osmotic pressure. We don't want water to, kind of rush into the cell and kind of essentially cause the cell to kind of explode. But the components of the bacterial cell wall are different from plant cell walls. Plant cell walls are made of cellulose with carbohydrates. Bacterial cell walls are made of peptidic glycan, which consists of proteins and sugars, which you'll see below. Pepto is a protein, And if you remember back from biology or anatomy even, proteins are composed of amino acids, right? Just a couple, or not a couple, but a bunch of amino acids. And a glycan, like a glycose or glycogen, is a sugar, right? And it says, part of the structure involves a carbohydrate, okay? So peptidoglycan is huge in bacteria, right? Because peptidoglycan is only found in bacteria, right? We don't have peptidoglycan in our cells as eukaryotes, and this is fabulous, and I'll get to that in a second. But it says anything that destroys that cell wall will cause lysis and destruction of the cell. So any way we can destroy peptidoglycan, right, that's essentially going to help us attempt to kill bacteria. And there are some enzymes out there that digest peptidoglycan. Penicillin works that way, where it kind of stops the formation of peptidoglycan. So as the bacteria is starting to reproduce, peptidoglycan is not able to be formed. And this is great because it's leading into this question down below where it says, what effect will chemicals that interfere with peptidoglycan have on animal cells? It's not going to have any effect on animal cells because we don't have peptidoglycan in our cell walls, right? We're eukaryotes. So if we're using a drug to kill a bacterium that targets peptidoglycan, it's not going to harm us in any way, shape, or form because we don't have peptidoglycan, right? Well, it's less likely to harm us, right? Go into that more later. So it's a great thing, right? It's going to be less harmful for us. So that's why if you can figure out how to target the peptidoglycan for that specific bacterium, you're more likely to have kind of a positive effect or less side effects, essentially, when you're treating your patients. This gentleman here, Hans Christian Gram, he came up with this method of Gram staining. We'll start with colors first, and then we'll talk about why those colors are there. But Hans, he discovered that Gram-positive microorganisms will stain purple, and Gram-negative will stain pink. Sometimes we say red, but definitely know Gram-positive is going to stain purple, positive purple, and negative tends to stain red or pink. Okay. And he uses those 4 chemicals we saw in the last chapter. If you remember when I said Cooper is a sweetheart, he uses that crystal violet, that iodine, alcohol, and safranin. Now, for a microorganism to be gram-positive, the reason why it stays purple is because it's got this very thick, look at this brown area right here, this very thick peptidoglycan layer. Okay, and the crystal violet sucks up that peptidoglycan layer, or the peptidoglycan layer, sucks up the crystal violet, just soaks it all in. And it's hard to get the crystal violet off because it's been absorbed. We utilize iodine to help it stay. But if we are gram-negative, our peptidoglycan layer is much thinner. Look at this brown layer down here, very, very thin compared to this thick brown layer right here, right? So the peptidoglycan, doesn't necessarily stick because it's so thin. It also has this outer membrane layer here, which is different, okay? This outer membrane layer is composed of a lipopolysaccharide. That's important. You really want to know that gram-negative bacterium, a major difference besides having a thin peptidoglycan layer is the fact that the outer membrane is composed of a lipopolysaccharide. This membrane becomes toxic when the bacterium die, and if we're exposed to it, it releases some toxins, okay? But the crystal violet's not able to stick on this peptidoglycan with the help of the iodine and the alcohol. It gets washed away, and safranin is then absorbed here, and this is how we get our pink or red color down here. So make sure you know your differences between gram-positive and gram-negative. Positive is purple, okay? Gram-negative is pink or red. Gram-positive has a thick peptidoglycan layer. Gram-negative has a thin peptidoglycan layer. Gram-negative also has this outer membrane right here, okay, which is composed of that lipopolysaccharide. And here's kind of it's not the best picture, but I really like it because I think it gives you a kind of a good demo or a good visual of the layers. You get this purple peptidoglycan layer here, thick for gram-positive, and then a thin peptidoglycan layer here, which is like a pinkish color, right, for gram-negative. And it also has this outer membrane here, this lipopolysaccharide membrane. So I do definitely enjoy that picture. All right, last slide for this video. I know it's a little bit of a longer video. I apologize. So what about what about bacteria without cell walls? So most bacteria have cell walls, but some have a cell, some may lack it, or some may have a composition that differs. Mycobacterium and tuberculosis is a good example. They have a very waxy cell wall. And we've talked about this previously, if you look at this last point down here, with the acid-fast staining method. So sometimes we have to use different staining methods because they show up gram-variable, both positive, and negative, which is what we're seeing here in this picture. And we can use the acid-fast staining mechanism to do that because of the waxy coating that the Mycobacterium produces, right? So we just kind of go about different ways of staining if we don't have that cell wall that we need. So it says some cell walls are composed of polysaccharides, and in others they are proteins. They will not gram stain, and at times appear gram variable, which is what we're seeing. And that's how we can use that acid-fast staining method or some other staining method to help us look at that bacterium. And that's all that I have for this video.